All right, here we have chapter 7 on the cellular and any type of membrane, its structure and also how it functions. This is Campbell's biology. So the most important overall function is that it provides a selectively permeable membrane for whatever environment it's blocking out, the cell or the nucleus or the Golgi or what have you. So only certain substances can pass across and others are prevented. Now it's made up of phospholipids, which are superabundant, and phospholipids are amphipathic. Just like an amphibian lives in both water and on land, amphi means both, so it contains both hydrophobic or nonpolar and hydrophilic or polar regions. The inside is hydrophobic and the outside is hydrophilic. It's often described as a fluid mosaic model, or that's how it's understood, where the membrane is fluid and constantly movable, and there's a mosaic of proteins passing through and stuck onto the surface. We've chemically analyzed these membranes to identify what it looks like, and we see here these phospholipids. Here's 2 fatty acid chains. In lieu of the 3rd fatty acid chain that would have made this a triglyceride, we've incorporated a phosphate group. And this phosphate group's attached to the glycerol makes this end hydrophilic, and thus it sticks out into the water. So if I were to take phospholipids and just drop a bunch into a beaker of water, they would form bubbles or spheres of a double membrane where the inside hydrophobic tails are pointing to the interior. Now, we didn't always understand exactly how the membrane was stacked. We knew it was made of protein and phospholipids, but we didn't understand the arrangement of those. So originally we had the Davson-Danielli sandwich model, which electron microscope pictures actually supported, and it said that the membrane itself was like a sandwich. The bread was protein, and the meat inside was the phospholipids, the fats. Today we know better because 1972, Singer and Nicholson, they won the Nobel Prize, and they proposed that the proteins were inserted in various regions on top and through the phospholipid bilayer. And thus this is the hydrophobic region of the protein, and this being the hydrophilic region of the protein. This should say hydrophilic. This is incorrect. This should be hydrophobic. Freeze fracture studies of the plasma membrane supported this fluid mosaic model from Singer and Nicholson, where they froze a cell, they fractured it and pulled it apart in half, and they saw these little bumps sticking out in the cytoplasmic layer, and the extracellular layer had fewer, but also some, as you can see there. So that freeze fracture studies, we now know, is our currently accepted hypothesis of membrane structure. Now, these phospholipids found within the membrane, they can move, and in fact, they move all the time. Here, these two might flip-flop left and right on the order of 10 to the 7 times per second. That's ridiculous. They can even flip-flop between sides once per month. That, of course, seems more likely or more logical, but these things move all the time. They're interchangeable. And we also add more phospholipids in all the time, and we remove them out as things are exported through exocytosis, for example. The proteins found within the membrane, we also know that they move around. This was some original research done using plasma membrane proteins between a mouse cell and a human cell, and they were radioactively labeled. And they hybridized, they combined these 2 cells together, and then they looked after 1 hour at how the proteins were arranged on this hybrid cell. And what they found was when they mixed it, some of the membrane proteins moved sideways within the plane of the plasma membrane, and they all sort of intermixed. And thus we knew that proteins moved around in the plasma membrane. Now, the chemical structure of the fatty acid tails of the phospholipid can affect the membrane fluidity because the more unsaturated a fatty acid, meaning the more double or triple bonds that exist, the more kinks that will occur, and as a result, these phospholipids are farther apart, and thus the membrane is more fluid. Compare that to saturated fatty acids. This one's a little bit more viscous. It has a harder time moving. It's a little thicker, if you like. Now, cholesterol is a steroid, and it is found within the plasma membrane as well. We often talk about cholesterol in negative terms. Cholesterol is bad, don't eat it, it causes heart attacks. Well, here we're talking about cholesterol in positive terms. It's found throughout all different membranes, all plasma membranes, and it aids in maintaining the membrane fluidity at different temperatures. The simple way to think about it is that the cholesterol molecules separate these phospholipids, sort of like the kinks did, but to help maintain the fluidity of the membrane. And because cholesterol reacts or behaves differently at different temperatures, this membrane will, of course, also react differently at different temperatures. The membrane itself here are integral membrane proteins. That means they pass all the way through. They are transmembrane. The core is hydrophobic and the outside is hydrophilic. We also have peripheral proteins, they are attached to the surface of the membrane and they stick out. These, of course, are transmembrane proteins, not peripheral proteins, but the proteins themselves are used for a variety of functions, 6 major ones that we describe here. The picture you see on the right, these proteins are used for transport. We'll talk more about this later. This one here showing active transport, this one here showing facilitated diffusion, both involving transporting of molecules across the membrane selectively. Here we have enzymatic activity. The enzyme is stuck in the membrane, whether we're talking photosynthesis or respiration or a variety of other reactions. Enzymes are often found stuck within the membrane as integral membrane proteins, and they catalyze reactions on the cell's interior, as you can see occurring here. Signal transduction is a way of carrying a signal from outside the cell to cause internal changes within the cell without that molecule actually passing in. So usually this is the result out here of a large hydrophilic ligand or molecule. Because it is large and hydrophilic, it has a hard time passing through the hydrophobic core of the membrane. Thus, it's forced to bind to a protein receptor, that is an integral protein receptor, and by doing so, it causes a conformational change or a shape change to this protein, and thus to a series of reactions internally as well. We will learn more about signal transduction at a later date. Fourth one is cell-cell recognition. Here you see a glycoprotein sticking out that enables this cell to identify this cell. This is fairly typical, of course, with the immune system and cellular identification and so on. Intercellular joining, we learned of various types of junctions previously, like gap junctions or tight junctions, desmosomes, and so on, where they allow adherence of one cell to another or passage of nutrients as well. And then lastly, we have the ECM and attachment of the cytoskeleton to the actual cell itself. Here we see the cytoskeleton internally. It's attached through this protein to the membrane, providing structure, and also the adherence of the ECM or extracellular matrix to the membrane itself. That's also done through proteins within the membrane. Here this talks more cell-cell recognition, carbohydrates used in that cell-cell recognition. And membranes look different on the different sides. We saw in the freeze fracture studies how the cytoplasmic side looked different from the extracellular side. This is the case, and this is due to the movement of proteins through the endomembrane system. That means from the nucleus to the ER to the Golgi. That's the endomembrane system, and thus affecting how proteins are synthesized. Here you see that occurring. Here is the ER passing these proteins through vesicles into the Golgi, and then from the Golgi another vesicle forming, merging with the membrane because they're all just phospholipids. Through exocytosis, the proteins go out. Only proteins who are made on ribosomes floating in the cytoplasm or cytosol, only those are destined to stay in this cell. All those that go into the endomembrane system are passed and secreted out. On this note, we will take a break.